And we have COVID-19, the theme of our brown bag. And today we have two presenters, Jesse and Karishma. So folks, you may share your screen. And we have a new folk in the audience today, Williams Jr. So let's all share.
Okay, so I am going first.
Oh, Karishma then.
Yeah.
Okay, let me.
How do I minimize this thing? Yeah, okay. Laser pointer. Okay, so hello everyone. Am I audible and can you hear me clearly?
Yes.
Yes, okay, great. So before I start, I want to thank all of you for being here today for my virtual brown bag. Today, I'm going to be talking about two papers which have shown similar experiments on how SARS Cov and SARS CoV-2 3A protein causes cell death. The reason why I chose these papers is because I work on understanding how cell death is regulated during the normal development of Drosophila. And I thought this might be a good fit and just a different perspective on how the coronaviruses can hijack the cell death machinery. And also, we always think of how the infection takes place and how the virus gets inside cells, but not how it really leaves the cells. So hopefully I should be able to pique your interest with this. And wherever you see these green ugly smileys, there is either a scientific or a fun fact associated with it. So let me begin by introducing the family of coronaviruses. So coronaviruses belong to the order Nidovirales and family Coronaviridae. They're further subdivided into alpha, beta, delta, and gamma coronaviruses based on the structure of their crown or the corona or the envelope spike. While the delta and gamma coronaviruses only affect animals and birds, the alpha and beta coronaviruses can be transmitted to human beings. What we have been dealing with since the beginning of early 2000s belong to the genus beta coronaviruses. In 2002, we had the SARS Cov pandemic, which affected 29 countries and caused around 900 deaths globally. After that, in 2012, the MERS Cov affected 27 countries and took around 900 deaths. 7 years from then, today, we're yet again faced by another member of this family, the SARS-CoV-2, which causes COVID-19. This virus has caused more than 900,000 deaths globally, and we're still counting. So it becomes all the more important to understand how these viruses work. So let's talk a little bit about the genome of these viruses. I was quite surprised by the fact that their genome is one of the largest genomes amongst viruses, which is about 26 to 32 kilobases in length. That's pretty large. They're non-segmented, single-stranded RNAs with a positive sense, which is the same as our eukaryotic mRNAs. So they also have a 5' end and a 3' end, along with poly A tails towards the 3' end. They contain multiple open reading frames or ORFs. ORFs are nothing but sequences that have transcription start sites, so they can be readily and quickly transcribed and translated. So 2/3 of this 5' end is covered by ORF1A and ORF1B region. And these are transcribed into two large polypeptides, PP1A and PP1AB, that code for 16 non-structural proteins, including the replicase and the transcriptase complex. The other 1/3 towards the 3' end of this genomic RNA is more or less conserved and is transcribed by the replicase and the transcriptase complex into subgenomic or shorter RNA fragments, which then code for several proteins, structural proteins and accessory proteins like the spike protein, the envelope, membrane, and the nucleocapsid protein. So one of these proteins, which is the 3A protein, is conserved from the SARS-CoV to the SARS-CoV-2 virus. And this is involved in apoptosis or cell death. So let's look at more detail into this 3A protein and how it causes apoptosis or cell death. So this first group, the first paper that I'm presenting in 2009, looked at the protein 3A for conserved sequences and found 3 clusters that are conserved. 3 cysteine residues, a motif containing a tyrosine residue, and a third region containing glutamic acid and aspartic acid residues. So to understand how important these regions are in causing cell death, they mutated these regions via site-directed mutagenesis. So cysteines were converted to serines. From here onwards, they're referred to as 3ACS. So wherever you see 3ACS, those are the cysteine mutants. The tyrosine converted to alanine. Here it is referred as 3AYA. And the aspartic acid and glutamic acid were converted to alanines, referred to as 3ADE. So they created these mutant proteins, and then they transfected African green monkey kidney cell line, which is Vero E6 cell line. And they also transfected that using the wild type 3A protein. All of these constructs are tagged by GFP so that they can visualize the subcellular localization of these proteins in the Vero E6. So they used this to study the localization, and the graph represents the quantification of the localization. So what we're looking at is that while the wild type 3A protein localizes on the plasma membrane, the mutants fail to localize to the plasma membrane and are mostly cytoplasmic. So the next thing they looked at is nuclear condensation as a marker for cell death. So basically, when a cell is dying, the nucleus and chromatin starts to fragment and condense as a result. So what they noticed is that when they overexpressed the 3A wild type in these cells, there's a high amount of nuclear condensation. That means there's a high amount of cell death taking place, which is reduced in their 3A triple mutant. So they checked if this is dependent on caspase activity. So caspases are nothing but proteins involved in several developmentally regulated processes. One of the key processes that caspases regulate is cell death, and that is also called as apoptosis. So they used a general caspase inhibitor to inhibit caspases. And what they noticed is that compared to the wild type levels of nuclear condensation, when a general caspase inhibitor is used, the amount of cell death reduced. What this suggests is that the SARS Cov 3A protein induces apoptosis and is dependent on caspase activity. So the next obvious question is which of these caspases are involved? The major caspases we know today that are involved in cell death are caspase 8 and caspase 9. Upon receiving a cell death stimulus, the cell activates these caspases, which in turn can activate other proteins of the cell death pathway and eventually lead to cell death. So what they found was that while the wild type has high levels of caspase 8 and caspase 9, the mutants themselves have lowered levels for these caspases. Now the thing to notice here is that not all of these levels of caspases are lost. So let's just keep that in mind for the next slide. So all of that was in vitro data, and they wanted to look at in vivo data to see what happens in cells. So they expressed their 3A wild type protein and the mutated protein in the adult eye of the Drosophila using the GMR Galfo driver, which I'm not going to get into detail about. What you need to know is that the wild type eye of Drosophila has this really beautiful distribution of a hexagonal array of ommatidia with an even distribution of these bristle cells. The thing to notice is that the distribution is severely disrupted in the wild type 3A and also in the mutants. What this suggests is that even if one of these sites is mutated in the 3A protein, there is still residual caspase activity from what we saw in the previous slide. And there's also cell death occurring. I was kind of wondering why they did not do a triple mutant study to show if it reverses these effects. I thought that would be interesting, but there's no mention of that anywhere in the paper. So the take-home message here is that these sites are important for plasma membrane localization and also for causing cell death, which is dependent on caspase activity.
Karishma, can I ask you a question? This is Anand.
Yeah, Dr. Sen.
This is really interesting. So in the cell lines, they use the triple mutant, but here they didn't use it.
Yeah, yeah.
And even mutating one of them is showing a range of cell deaths. So that means you need to mutate all 3 sites for it to restore, or otherwise, even if these 3 are mutated, you will still see leaky cell death phenotypes.
Yes, so they haven't mentioned anything about the triple mutant in their adult Drosophila. And I don't know if that survives or not, but they haven't said a word about it.
Okay, thank you.
Okay, so that brings me to the last data set from this paper before I move to the next paper. So the 3A protein back from 2006 in a study is known to be a sodium-potassium ion channel. So here this group wanted to study if the ion channel activity is somehow linked to its apoptotic activity. So they did an electrophysiology experiment using a whole cell clamp patch. They transfected their HEC293 cells, which are human epithelial kidney cells, with 3A and the different mutants tagged with GFP. And based on the GFP expression, they picked these cells and applied a regular voltage from -100 to +100 millivolts with a 20 millivolt increase. And they recorded whole cell currents from that. So the extra thing they did in this is they applied barium, 10 millimolar barium. So barium blocks or inhibits all ion channel activity. So you would expect that in the wild type, the membrane potential to either drop or not change much with the barium application. And that is what we see with the wild type 3A protein. But in the mutant proteins, there is not a significant change in the membrane potential from before and after the barium application. So if you remember from the very first data set that these conserved regions are important for plasma membrane localization, it makes sense that if these proteins are unable to localize to the plasma membrane, the membrane potential would be low. So the take-home message from this slide is that the ion channel activity of 3A is important for its apoptotic activity. So before I move into the next part of my talk, are there any questions? Okay. So this next group that did a similar study was run et al. 2020, but they used the 3A protein of the SARS CoV-2 virus, which causes COVID-19. So they began by showing the cell death activity of the 3A protein transfected into 3 different cell lines. So they've used human epithelial kidney cells, human liver cancer cell line HepG2, and again, the African green monkey kidney cells, Vero E6. The cell death is detected by 2 dyes, propium iodide and the annexin V-FITC. And both of these dyes marks the fragmented DNA, so it goes and binds to any DNA that is breaking down. And the recording is done using a flow cytometer, as you can see. So compared to the empty vector, there's an increase of the cell death signal in all of these cell lines that have been transfected with the SARS-CoV-2 3A protein. And they plotted the percent apoptosis of that, and this is pretty easier to understand. So wherever these cell lines are transfected with the SARS-CoV-2 3A, there's an increase in apoptosis. So 3A is causing ample amount of cell death here. And interestingly, they also noticed increased level of caspase-3. Caspase-3 is a very important feature of caspase-dependent cell death. Only after caspase-3 activation, the cell really goes into the dying mode, which is accompanied by nuclear condensation, DNA fragmentation, and membrane blebbing. So they also looked at other markers for cell death using some Western blots. So at 24 hours, they saw increased levels of truncated bit protein. They saw BCL2 and then cleaved caspase-8 and cleaved caspase-9. They also saw increased levels of cytochrome C leakage. So all of these proteins are involved in the cell death process and are required for deactivation of downstream caspases. Additionally, they also noted that the membrane fraction for the SARS-CoV-2 3A transfected cell lines contain the sodium potassium ATPase activity, which resonates with the first study that I presented. So the take-home message here is that the 3A protein of the SARS-CoV-2 activates cell death via increased levels of all of these cell death proteins, and it also has the sodium potassium activity. And that brings me to my last data set from this paper. They've done a really nice comparison, I thought, of cell death levels and caspase-3 levels here in the SARS-CoV-3A and the SARS-CoV-2 3A. As you can see, the levels of apoptosis and caspase activity are lower in the SARS-CoV-2 as compared to the SARS-CoV-3A. They said the reason in the differences in the levels of cell death could be due to the difference in their pathogenicity. And we also know that the SARS-CoV-2 is less virulent than the SARS-CoV. And that is also the reason why people with SARS-CoV-2 can be asymptomatic. So I thought this was a really nice data set. So to conclude all the findings, the ORF3A codes for protein 3A, which has conserved domains from SARS-CoV to SARS-CoV-2. The conserved domains are important for the plasma membrane localization of the protein, and that can lead to disruption of the membrane integrity, nuclear condensation, and mitochondrial response and cytochrome C release, and activation of several other cell death markers. This in turn activates the downstream caspases such as caspase-8, caspase-9, and caspase-3, which in turn lead to apoptosis. And the level of cell death is lower in the SARS-CoV-2 than the SARS-CoV. And this is how the coronavirus hijacks the cell death pathway. And I also put a tiny little video for you guys. So
this is SARS-CoV-2. It belongs to the family of coronaviruses named for the crown-like spikes on their surfaces. SARS-CoV-2 can cause COVID-19, a contagious viral infection that attacks primarily your throat and lungs. What actually happens in your body when you contract the coronavirus? What exactly causes your body to develop pneumonia? And how would a vaccine work? The coronavirus must infect living cells in order to reproduce. Let's have a closer look. Inside the virus, genetic material contains the information to make more copies of itself. A protein shell provides a hard protective enclosure for the genetic material as the virus travels between the people it infects. An outer envelope allows the virus to infect cells by merging with the cell's outer membrane. Projecting from the envelope are spikes of protein molecules. Both a typical influenza virus and the new coronavirus use their spikes like a key to get inside a cell in your body, where it takes over the cell's internal machinery, repurposing it to build the components of new viruses. When an infected person talks, coughs, or sneezes, droplets carrying the virus may land in your mouth or nose and then move into your lungs. Once inside your body, the virus comes in contact with cells in your throat, nose, or lungs. One spike on the virus inserts into a receptor molecule on your healthy cell membrane like a key in a lock. This action allows the virus to get inside your cell. A typical flu virus would travel inside a sac made from your cell membrane to your cell's nucleus that houses all its genetic material. The coronavirus, on the other hand, doesn't need to enter the host cell nucleus. It can directly access parts of the host cell called ribosomes. Ribosomes use genetic information from the virus to make viral proteins, such as the spikes on the virus's surface. A packaging structure in your cell then carries the spikes in vesicles, which merge with your cell's outer layer, the cell membrane. All the parts needed to create a new virus gathered just beneath your cell's membrane. Then, a new virus begins to bud off from the cell's membrane.
So this was just exocytosis. I couldn't find a video which actually shows like 1 million virus particles bursting out of a cell, but I thought that was interesting. So with that, I'd like to thank everyone, my faculty mentor, Dr. Kanga Singh, my committee members, the Department of Biology and the University of Dayton for the funding, and Singh Lab members and colleagues. And with that, I'll take questions.
Any questions for Karishma?
So, Karishma, I have questions. So this is very interesting that it hijacks the cell death machinery, and but in what stage of infection this machinery is hijacked, because it has to replicate and then get released. So what do you think that's during the course of infection, what stage might be this thing happening in the cell?
I think it's probably the last stage when all the virion assembly has been taken place and all the particles are assembled and ready to move out.
So it is not at all using the nucleus of the host cell. It goes stays outside only.
Yeah. So I found some videos with other videos that they have shown that it only stays between the interspace of the ER and the Golgi complex.
Oh, okay. So it has less restriction of movement.
Right. So I don't think it undergoes like a lysogenic cycle where it goes and like gets into the DNA of the host. I haven't come across any studies like that because viruses usually go through lysogeny or the lytic cycle, right?
Yeah. So this is really interesting thing happening.
Yeah.
Hi, Karishma. That was really great. It was very clear.
Thank you, Dr.
I'm wondering, because both of these papers use transfection, right? Did they mention any studies where they actually use virus infection to see if the 3A has any mutations, the viral load would be lower or higher in a cell?
No, they haven't done that. And I was kind of wondering if they could at least like use some lung cell lines or something like more related to, like the throat but they haven't done any of that. They haven't even used any patient samples or. Does that answer your question?
Hi, Karishma. I have a question. This is Madhuri. So they do this by using transfections, but when they do it in Drosophila, are they able to recover any parts of the protein that they can then, did they? So basically my question is, for all the mutant proteins that they created, did they put them back in the virus and then see if there was a comparison of how these mutant proteins behaved? And
that would be interesting, but no, they haven't done that.
So these were all transient transfections and they could not recover things back and then sort of serially study what happened.
No. They did not like even make any lysates or anything from Drosophila.
Okay. Thank you.
Great, thank you, Karishma. It was a great presentation.
Thank you.
And now it's Jesse's turn. So Jesse, you may share your screen. And take the lead.